We good? Welcome back. Okay, so I think this is a good idea, and you guys tell me if you agree with me. I think sometimes my videos are a bit too long. I think sometimes when you're sitting there for 2.5 hours, it gets difficult, right? And I want to make a quick note here. On the topic of advertisements on my videos, many people message me and they say, dude, I love your videos, but there's so many ads. I will tell you a story. When I first monetized my YouTube content, I went on there and I said, you know what? Let me turn on the monetization, right? And the reason that happened is because someone reached out to me before, a very close friend of mine actually. He said, dude, I was watching one of your videos and an ad came up. I didn't know you get paid. And I was like, I don't get paid. And I was like, how many ads come up? He's like, maybe once every 10 minutes or so. I was like, that's pretty frequent. And I had no money out of it. And I had been uploading to YouTube for 6 months at that point. So 6 months, thousands of views, hundreds of thousands of hours, no compensation, filled with ads. And I ran an experiment. I turned my ad revenue on, and I asked my friend, I was like, go watch one of my videos again. Did the number of ads go up? He said no. So the only difference was that I was making money now. So regardless of whether I make money or not, the ads will be there. So why wouldn't I make money? Why wouldn't I monetize this? You can also pick which ads you want on and off, because certain ads are haram. Alcohol, gambling, dating, that one thing I can't mention on YouTube. You can turn on and off these, ads. So all I had to do was turn off everything that I don't agree with. Halas. And that's how I pay my rent. I'm being very serious. YouTube is my rent money. I'm being very, very serious. Being very forthcoming with you guys. So thank you guys for paying my rent. Let's keep studying. Where were we? For the people who had to leave for a little bit to wash up before prayer, I want you to look at the board. We were talking about the different, roles of the smooth and the rough ER, right? Smooth and the rough ER. And I said that the smooth ER was like detoxification, not really started with ribosomes, not started with protein factories, things like that, blah, 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 whatever it is, right? And the rough ER is more so studded with all these protein factories and blah, 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 and this and that is continuous with the membrane, et cetera, et cetera, et cetera. I asked a question. I said, if I looked at a hepatocyte, a liver cell, right? And I looked at a myocyte, a muscle cell. There's a ladybug on this eraser. Oh, it's so cute. Look at that. It's adorable. You guys see that in the front? Very cute.
You just noticed it?
Huh?
It's been there?
Yeah. Has it?
Yeah, it was going all around.
Really? That's adorable. I love ladybugs. I loved them since I was a kid. There used to be a bunch of them in my garden. I used to sit down. They used to crawl in my hand. Right? Lots of ladybugs on Long Island. Lots of bunnies at night. They hop around in the streets. Right? Lots of raccoons too. They're not so nice. Right? Lots of geese, lots of raccoons, lots of ducks, lots of bunnies. It's an island, Okay, anyways. If I took a muscle cell and a liver cell, I compared them, the relative presence of the smooth versus the rough ER. We need to know that different cells have different sorts of components in different compartments in different concentrations in different amounts to do different jobs. The job of the liver is first pass metabolism, like I said before, where when you eat things and you digest them and they drain through your GI system and they go through the portal vein to the liver, they get metabolized there. And that helps with digestion and this and that. Demon Slayer is a very good show. Very, very good show. I read the whole thing while I read the last couple chapters of Demon Slayer while I was writing my philosophy thesis. I would write a page of my thesis and then read a chapter of the book. And I did that until 6 in the morning. I started at 11, I did till 6. Because I wrote 24 pages of my thesis in one night. Okay. So, this compartmentalization, this sort of functioning, is different across the whole body. So obviously, cells are designed to sort of do different jobs, do different roles. This differentiation, specification, everything. Determination, differentiation, specification, we're going to talk about all this, right? Because I need to look at my notes. I honestly don't remember. I'm not a cell bio person, right? To be honest, are you guys enjoying it? Put your hand up if you're enjoying this. Are you guys having fun? Okay, fantastic, amazing, right? I hate this chapter. This chapter sucks. I hate teaching this chapter. So you guys are having fun in a lecture that I hate teaching. That's a good sign for me. That's a bad sign for you. Because that means when I love teaching a lecture, it's not only going to be more enjoyable than this, but it might also be a bit more difficult. Because I'm going to be able to explain more and elaborate more and do more and this more and that more. However, I will set a rule very early on. If you need me to slow down, please speak up. The value of one student's opinion is worth more to me than my entire video. If I need to re-explain something that takes 10 minutes to re-explain, I would rather do that than please everyone else in the room. So if you need something re-explained, tell me. I am very sincerely asking. Okay? Because if you leave out of here and you don't understand it, it's my fault. It's not your fault, it's my fault. Okay? And I don't play that. I don't accept that for myself. Okay? And if you don't want to ask in front of everyone, come ask me personally. Okay. Does everyone understand that? Does everyone understand the point I was trying to make here? Very good. Okay. Does anyone have a cool physiologic or anatomical abnormality about them that they're not uncomfortable sharing? I, for one, am colorblind. I am colorblind. I have externally rotated hips, meaning my feet point out like this when I stand. They don't point forwards, they point out. If I stand like this, my hips are rotated inwards, and my knees are knocking. I have to stand like this to be comfortable, right? And I have a herniated disc.
Hypoextended elbows.
Wow, that's cool. Almost double jointed or what?
No,
Or like fully double?
No, not hypo.
Oh, hypo.
Yeah.
Interesting.
Doesn't go fully 90.
Interesting. That's cool. Anyone else?
My optic nerve is bent.
Your optic nerve is bent. Does that affect your vision at all?
No.
Interesting. How do you find that out?
I volunteered for the class presentation, and then my teacher checked, and he goes, "Wait a second, it's bent."
Wow, that's cool. If you ever get an MRI done, you'll be able to see it. That's cool. MRIs are scary. Anyone ever been in one? I had to go for one when I herniated my disc. It happened during my OBGYN rotation. I woke up one morning, I got out of bed, I planted my left foot, and I just fell to the floor. Like pain. Like pain, burning, radiating, shooting, like all the pain you can imagine. Anyone ever herniated a disc before? Anyone? Okay. It's painful. It hurts. But alhamdulillah, I'm better. Doing a lot better. Toradol's one hell of a drug. Toradol's nice, man. Listen. Toradol's nice. Because do you know what Toradol is? It's an NSAID. It's like really strong Advil with a tiny, 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 tiny little bit of heroin. That's Toradol. Toradol's cool. All right. Cooler things are coming. Because now we're talking about the Golgi apparatus. Oh my God. Stupid. It's a New York classic. Whenever I got mad at a marker, I'd toss it across the room because the garbage can was over here. Yeah. Huh? It's on. Yeah. I can see it. You guys hear me? I'm not using the class microphone, but you guys can still hear me, right? I am a very loud person. I outshouted a protest one time. I outshouted a protest. Okay? Have you guys been to New York City?
Yeah.
Have you guys been on like Fifth Avenue with all the skyscrapers and everything? When I shout, it echoes off the buildings. Yes, I'm a loud person. Golgi apparatus. Probably one of the coolest parts of the cell. Probably. Is this anyone's favorite part of the cell? Everyone has one. This is my favorite part of the cell. Whose favorite part of the cell is the mitochondria? You're so lame. You're so lame. You're either lame or you're a gym bro, like, or both. Definitely both.
I did it for a 7th grade project once, and ever since then.
Makes sense. You ever seen balloons when they're deflated and you stretch them out and there's like this little bit of space inside? and you sort of like push your finger through it, that's exactly what happens with the Golgi apparatus. It's a stretched out membrane like this, and it involutes on itself. So imagine a sphere, like a soccer ball, and you flatten it, you stretch it out. There's a tiny bit of space on the inside. And basically what you're doing is you're pushing stuff through it, and a bleb forms. And now you have a smaller sphere made of whatever the Golgi was made of, and it carries what you push through it. Does that make sense? Yes? Guys, does everyone get that visual? Yes? If not, tell me now.
No.
No? Okay. You have a membrane. And as you push through the membrane, what happens is this membrane goes like this, and then goes like this, and then goes like that. And as you pushed through it, you ripped a piece off of it. And that piece now surrounds whatever you push through it. Does that make sense? Yeah. So think about, here. Push, push, push. And imagine I can push my whole finger through here until, pop, it comes out with a little piece of the tissue like that. That's what the Golgi does. Okay. So the Golgi apparatus is a series of membrane-bound sacs. Don't laugh. Everyone started laughing. Damn it. My mom was right. I am stupid. My mom thinks I'm brilliant. And every single time she says that, I'm just like, oh, I'm so sorry to disappoint you. I'm so dumb. Guys, you can be very academically intelligent and be a very dumb person. There are some very dumb people in medical school. I'm one of them. It holds the job of modifying synthesized proteins. Yes? Modifying synthesized proteins. I put modifying in quotes. Why? That is a whole discussion in and of itself. Post-translational modification are words that shake me to my core. I hate those terms. Anyone here ever had surgery done? For anything? Anything at all? Do you guys mind sharing? ACL? Sorry? Appendix? Adenoids, okay. Post-translational modification is cellular surgery, molecular surgery. It is precise. It is exact. It is only done when necessary. And it completely changes you for the rest of your life. Okay? Guys, one more thing that I go on a rant about all the time. Plastic surgery. Trauma plastics, burn plastics, reconstructive plastics, lymphatic plastics, some of the coolest, most amazing, cutting-edge science the world has ever seen. These people are lifesavers. When you can give someone with cancer who has their lymph nodes destroyed proper drainage from one part of their body so that they no longer need to feel heavy and gross and get it drained and like have their literal limb rotting off of them, when you can do that or reconstruct an acid burn victim's face, you deserve respect. However, I am not going to give over here, now over there, over here, in the front, now on the camera. Okay. There's a reason I say that. There's a reason I say that. Because when you look at the phone screen, you tense your eyes. It's bad for you. It's very bad for you. You get a frontal headache, right? So, surgery is meant for stuff like that. Guys, nothing in this world is worth getting a unnecessary surgery over. I will leave it at that. I will not speak further on the matter, but those are my opinions as someone who's seen it happen to other people. There is no reason to get a surgery that is not medically necessary. Unless it is to fix a gross deformity or to bring you back quality of life, don't do surgery. Surgery is one of the scariest things a human being will ever go through. And one of my mentors who was an orthopedic surgeon said, when someone comes to a surgeon, they trust them with whatever life they have left to give them back the life that they want. That's when you get surgery. Okay? Yes? I will give you these little tidbits of information as time goes on. And I hope that it changes you, because it changed me. So, the Golgi modifies things and then also packages them. It packages molecules into vesicles. What are vesicles? Like these little membrane blebs that carry stuff throughout, the cell in order to get from one place to another, things like that, right? Not much else you need to know about that. And the vesicles actually merge with what? Those vesicles, they're little membranes themselves, they merge with what? The cell membrane, yeah. And these vesicles merge with the cell membrane. When my mom looks at my handwriting, she says it's like halfway between script and garbage. I hope you guys agree. You're allowed to agree. You're allowed to make fun of me. I make a lot of fun of you guys. You're allowed to make fun of me. I won't take it to heart. I'm just going to go cry when I get home, but I'm not going to take it to heart in front of you. Hey, no blame on you today. We have fun here, guys. We have fun. We learn, but we have fun. I hate boring professors. I hate it when people who are like, oh, I have been in the field of pediatric oncology for 35 years. And I was like, and yet you can't deliver a 30-minute lecture. You can't hold my attention for 30 minutes. How? I feel so bad for the people you interact with. Like, that's just so sad. Like, you're sitting here, 30 plus years of experience, I don't even want to listen to you. I don't want to listen to a word that comes out of your mouth. That's so sad. If I ever become like that, guys, of mice and men, right in the back of the head, please, please, put me out of my misery. So the Golgi apparatus. What's that album that's like the cover is like that, like sound wave, like this? And then... Aha! I got him. Someone check in on her. Arctic Monkeys, yeah. I don't listen to music anymore. I used to, so I remember that album from when I used to listen to music. And then it kind of reminds me of that, like the way that the Golgi is drawn, kind of reminds me of that, like this little sound wave. All right, guys. So, can someone put together a story here? Like, what are we looking at here? It looks very simple, but we are actually looking at the basics of not the hepatic portal vein. That, I might need to call on some friends to explain. I loved hepatology. It was cool. Very cool science. Did you guys know that without your liver, your blood doesn't clot properly? Your blood doesn't clot properly. You don't detoxify drugs properly. You can't make and break down amino acids properly. You can't regulate your sugars properly. Lots of other things. The liver does everything. Literally everything. Shout out to your liver, man. Right here. It's huge, by the way. It's like that big. In Bangla, if you really love someone, you call them a piece of your liver. That's what my mom calls me. You don't hear like eating liver? I love eating liver. Oh, so good. So good. Okay, this is the basics of the endomembrane system. And in order to describe the endomembrane system and talk about exactly what's going on, I need to write you guys a story. 1 second. I need to tell you guys a story. The DNA inside of our cells is turned into mRNA. By the way, guys, if you're thinking that we're doing a lot of talking today and like over-explaining this and this and that, it's not going to be like this every time. The first lecture is always like this. If you stick it out, promise, things go a lot faster. We are spending lots of time to build a strong foundation so that in chapter 7 of biochem, when you guys are drowning in your work and absolutely being suffocated by the work that you're doing, I don't need to re-explain how mRNA works. Okay? Stick with me. Sabrakaro, please. Right? mRNA then turns into what? Guys? Proteins. Loud. Okay? The proteins are synthesized where? Loud. Ribosomes. Good. Okay. These proteins are packaged into the vesicles from the loud. Good. And the Golgi, these vesicles will fuse with the membrane. From the DNA to the RNA to the proteins to the vesicles to the membrane. This pathway actually draws a straight line. We have DNA, it turns into mRNA. This mRNA hits the ribosome, gets turned into a protein. This protein travels up, hits the Golgi, gets modified, moves, gets packaged into a vesicle. That vesicle fuses with the membrane and exits out, and the protein leaves the cell. This is known as two things. Endomembrane transport, number one, through the endomembrane system. And number two, that last step is known as exocytosis. The process by which something inside the cell will leave the cell. And the way that that happens is this. Very interesting. You have the double membrane. When I draw a membrane, think double membrane, okay? You have the double membrane, you have a double membrane of a vesicle. Those two membranes are functionally what? The same. Say it. Functionally the same. As this membrane approaches this one, you get a closeness and a closeness until they touch. And remember, these membranes are functionally the same. So this membrane can actually open up into this one and open up a little bit. And the protein's in here. And when this membrane is pushing, it's able to just, can you guys imagine sliding these pieces out of the way? And it opens up, and this membrane becomes just part of that one, and the protein gets pushed out, like a little that. It goes, it fuses, and it goes, just becomes part of that cell membrane. Interesting. We said that they were functionally the same, so realistically and thoughtfully, it makes sense. It's logical that those membranes can fuse, but if we keep doing this over and over and over again for every protein that we make that's being sent out of the body, isn't the cell membrane just going to get huge? How do we compensate for that? This is exocytosis. There must be endocytosis, where the same thing happens from the outside. Guys, I have the picture up here for a reason. I didn't erase it. Imagine this pushing in, just like we did with the Golgi, ripping through it, and closing up, and closing up, and coming in. Exo and endocytosis. Okay? One of them has a vesicle from the inside that it sends out, and one of them takes the cell membrane and makes a new vesicle with the material, because the cell membrane and the vesicle membrane are one and the same. Fantastic. You guys are doing great. And yeah, I'm doing great. I'm having a great time here, guys. To be honest, you can ask them. I was very nervous before I came here. I have not taught in this fashion in a very long time. I'm very, very happy that it's going this way. And I'm glad that you guys are enjoying it. And even if you're lying to me, I'm glad that you're lying to me. I used to spend a little bit of time talking about peroxisomes. And honestly, not super important. But I do want you to keep in mind that hydrogen peroxide has a specific function within the cell. that hydrogen peroxide as a disinfectant, as an antimicrobial, as a radical that is able to sort of just absolutely wreak havoc on things inside and outside the body and things like that, which is why you pour hydrogen peroxide on a wound to get rid of all the stuff, all the bad stuff, etc. This is naturally antibiotic, antiviral, anti-everything bad. Peroxisomes can help with the destruction of those sorts of nasty compounds inside the body. And what is hydrogen peroxide? Okay, peroxide, right? Is anyone very astute at general chemistry? Anyone? Anyone? A chemistry whiz? Because I'll tell you one thing. I am. All right. Peroxide is O2 2 minus. Do not ask me how that happens. Do not. Okay, fine, I'll tell you. That's a breakup waiting to happen. That's crazy. That is nuts, right? That's ridiculous. Because both of them would want nothing more than to have a double bond right there, correct? Yes? You guys remember from general chemistry? What happens if this guy picks up a proton and that guy picks up a proton? Be very deliberate when you draw molecules. Be deliberate. Everything I do is for a reason. Is their problem solved? Not really, because that tug of war on the middle bond is still happening. Oxygen single bonded to oxygen is like, they're playing at it. They really want to rip that thing apart, right? And eventually it does happen. Single arrow, not a double arrow. What do I mean by double arrow? This is a double arrow. This is a single arrow. It's called a fish hook, right? The difference is huge. This means one electron. And big electron bubble right there. 2 hydroxide radicals that can rip almost anything apart. Okay. Guys, I got into a whole discussion about my students, about what type of bond dissociation this is. And they kept trying to tell me that I'm wrong, but I'm not wrong. I promise you I'm not wrong. This is called a homolytic bond dissociation. They're like, how is it homolytic? Electrons are going 2 ways. I don't care. Homolytic because it makes the same thing. A heterolytic bond dissociation makes different things. If I'm wrong, bring me evidence that I'm wrong. Okay. There is one part of this chapter that I'm not very good at. It is the cytoskeleton. Okay? And the thing is that I'm not good at explaining it, and that means it's a waste of my time to try to do it, because I'm not going to give you information that is poor. That's not what I'm here to do. And I am more than okay with admitting that. So, read up on what the Kaplan book has to say about the cytoskeleton. But until then, I will narrate you some things directly from my notes that I took away from it, from my understanding. Okay? The cytoskeleton. Basically for structure, shape, things like that, correct? We have microfilaments. We have microtubules. We have intermediate filaments. I can just hear the frantic tapping of Apple Pencils. It's so funny. Oh, I do not envy you guys. Imagine if I had to deal with me. Oh my God, I wouldn't last an hour. Seriously. You guys are troopers, man. Microfilaments are made of actin. One of my favorite museums is the MFA. The MFA, I've been in Boston, I've been in Philadelphia, and I've been in Houston. Is there one in Dallas? I don't know. The MFA. And what you need to know about them, actin specifically, is that it interacts with myosin, and this leads to muscle contraction. And it also plays a role in cytokinesis. Notice how I wrote that. Actin plays a role with myosin. Microfilaments have a role in cytokinesis. And microfilaments are made of actin. So that doesn't necessarily mean that microfilaments in and of themselves have a role in muscle contraction. It's much bigger than that, right? Specifically, they form the cleavage furrow in mitosis, correct? The cleavage furrow. What is the cleavage furrow? It's that thing in the middle where the cell begins to involute and split down the middle. What is that process by which it splits down the middle? What's it called? Huh? Good. Well, we had it on the board. I think there's another one, right? There's one more thing. I forget. We'll come across it. Too much medical stuff in my head. There's like, how do heart attacks happen? Right, actin. Microtubules are made of tubulin. And this is how kinesin and dynein, I can never spell this right. Kinesin and dynein move around the cell. Dynein dives into the cell, goes towards the cell center. Kinesin goes the opposite way. What's so funny? You want some peanuts? Okay. I'm hungry too, man. Actually, I'm not that hungry. Yeah, what's up?
I have a question for microfilaments. Since like the cleavage furrow, does it also act on like the cell plate implants, or is that just act?
That is a question for a cell biologist. That is a very good question, though. I want you to ask it to someone and come back and answer me. Hold on. Everyone on YouTube can hear me chewing. If you have a cell, and you have a cell center, and you have a railroad all around the cell, the protein that will dive into the cell will be dynein, and the one that walks out will be kinesin. So this way will be dynein, that way will be kinesin. Does that make sense? This is important when it comes to neurons. The cell body of a neuron may be all the way over here, and you have this long axon with the axon terminal. And I say that something's moving from here to here. And you may say, oh, we're going out from the center of the cell. Center of the cell's right here. No. Cell body's right here. That's still moving towards the cell body. I don't care if you're here or here. The cell body's in one place. Kinesin will move away from the cell body, and dynein will move towards. Is that understood? Cilia and flagella are also made of microtubules. Flagella are some of the coolest things ever. The fact that you have an entire motor, a generator, packed into the tail of a single sperm cell is nuts. That's crazy, man. Crazy. Not as crazy as pregnancy. I will consistently remind you guys that pregnancy is the coolest thing you'll ever study in medicine. It's sick. It's awesome. Secondary to, of course, cardiology, you know. Anyone here want to be an OBGYN when they grow up? Yeah? It's a cool field. Definitely made me question a lot of my decisions in medicine about whether or not I want to do it. But I decided there's probably better people than me to do OBGYN. I had one of my best patient interactions at OBGYN because our translators weren't working. And I had to do a full medical interview in fluent Bangla for an hour. It was a very, very, very life-changing experience for me and the patient. And she ended up coming back and asking for me 2 months later. I was at a different hospital. because no one else understood her. Speak to patients in your native language if you have one. They love it. I don't care who in your hospital tells you not to, or says you can't translate, or you can't do this, or you can't do that. If you don't do it officially, do it unofficially. Find the patient yourself, hunt them down, and speak to them in your own language. Okay. Intermediate filaments basically just help anchor organelles. By the way, I know we said until 9. We will more than likely go over time many times. If you guys want to leave now or later or at 9 or whatever it is, guys, I'm not going to be offended in the slightest. I am here to teach. I do not care how long it takes. I couldn't care less. If I get home at midnight tonight, I would think that this was worth it. Okay? If you guys need to leave, leave. However, every single time I've said that, if a student needs to leave, they've always apologized, which makes me feel very good because it means students like being here. But listen, that doesn't mean you can't leave. If you were gonna leave right now, please feel free. I'm not stopping you, I promise. Okay. There is a little bit more about the cytoskeleton, but nothing else that I really feel comfortable talking about because I'm not that great at this stuff. We're almost done, guys. We just have a little bit to talk about the different types of cells and then a little bit about the way that cellular organisms work. And that'll be it. Thank you guys for coming. Thank you so much. I am going to talk in tacos later.
I'll show up.
Text me.
I got you.
Okay. Tissues. Epithelial. Endothelial. Neurons. Muscle. How you doing? Keeping up? Good. How are you doing? I can't believe you guys are even here. Isn't that crazy? So weird.
You never know, man.
We're going to replace endothelial with connective. I don't love that, but I'm not the boss here. Actually, I am, but I don't know anything. Epithelial. Covers the body and lines its cavities. One time I was teaching, and I had a friend who was sitting in the lecture hall. I was like, I'm trying to line your cavities. That dude was hot. My friend's a good-looking guy. Huh? Involved in absorption, secretion, etc. I like it when students laugh. That's good. It helps you remember things. These things connect to the basement membrane, which is basically just the stuff that sort of connects everything together, which is why it's part of connective tissue. But more importantly, these are the cells that usually make up a very specific set of cells known as the parenchyma, the parenchyma, or the parenchyma, which is basically just the active portion of an organ. If you're talking about the liver parenchyma, it's the stuff that is able to do the stuff that the liver does. If you're talking about the brain parenchyma, it's the neurons and the tissues that generate the signals. If you're talking about the thyroid parenchyma, they make the thyroid T3, T4, the hormones, correct? You guys getting that? Good. There are numbers of layers, and you guys have learned this extensively. So I'm not going to draw the diagram like I normally do. We have simple, single layer, epithelium, right? We have stratified, multiple layers, and pseudostratified. So they're kind of laying on top of one another. They look like taller, shorter than one another, but it's not really like that, right? Does anyone know where in the body we find transitional cell epithelium? It's a trivia. So one specific part of the body where we find transitional cell epithelium. There is hey, he got it, the bladder. Yeah, very good. That is where transitional cell carcinoma comes from. Anyone here worked in a urology office? Ever? Or like been with a urologist? Anyone know urologist? Okay. Transitional cell carcinoma is a specific type of cancer that happens only in the bladder, and it usually happens in men. What's the number one risk factor of transitional cell carcinoma? Smoking! 50% of the time, you're right. Okay. Shapes? Cuboidal, columnar, squamous. Flat, cuboidal is a cube, columnar is like a column. Correct? I like this. I see people like leaning back, relaxing, laughing. It's good. This is the way class should be, right? We're supposed to have fun here. Who is the best teacher? If someone in this room thinks they have had the best teacher ever, tell me now. Tell me the story. Yes?
Bruce Novak for OChem.
OChem? Bruce Novak. What was special about Bruce Novak?
He was just like,
Taught us
just
math.
chill, like he made everyone feel comfortable. He explained things really well.
Good.
He didn't do PowerPoints. He like would handwrite
Yeah.
his notes.
Yeah, 100%. I feel like one thing that I've noticed about all of the teachers that I've personally really loved is that they crossed that barrier between the text and the student. It's almost like they were the thing connecting you two. You know what I mean? Because some teachers are just like, here's the text, here's the information, you're the student, right? And they just act as a translator rather than someone who is bringing you into that world. You know what I mean? Connective tissue. These guys make the framework for other things to rest on and do their thing on, right? And most cells in the connective tissue produce and secrete materials like collagen and elastin, which produces the extracellular matrix, the ECM. I have a nasty scar on my left leg. Gross. It's like, I don't know if the camera's gonna get that, but it's like that big. It used to be that big and now it's shrunk, it's like this big, but it's a keloid scar. And it's an overgrowth of abnormal collagen because my body was so violently reactive to what happened. So like you just had an overgrowth of all this collagen, got super disorganized and blah, blah, and this and that. But that's basically the stuff that lines your extracellular matrix. So now, if I scratch this part of my thigh, I can't feel it. I don't have any nerve endings in this part of my leg anymore. So I actually can't feel, I feel pressure, but I can't feel myself scratching my scar. What happened was I was running a 600-meter race behind my team captain in my junior year of high school track and field in the winter. And it was so cold outside that I actually didn't feel my legs. It was like 28, 29 degrees outside. And I had on a pair of very thin tights and shorts and a tank top because I'd taken off my sweater to race. He stops dead at 400 and his racing spike goes straight through my thigh. That's how I got that scar. And I didn't feel a damn thing. I didn't realize, racing spikes are basically like these little needles at the bottom of shoes that help you to grip the track a little bit better when you're racing. They're actually very helpful. But I didn't realize it at the time until I saw the trail of blood on the last 200 meters of the track while I was racing. And I looked down at my leg, and you can see the blood all down my calf. And I tore a nice pair of tights. It's unfortunate.
Did you win?
Huh?
Did you win the race?
Of course I did. What are you talking about? Okay. I don't love the next part of the chapter. They talk about like archaea and bacteria, and they go on this whole tangent. I took so many notes, and I color-coded them, and I did this, and I did that, all for them to be like archaea are like the in-between. They're kind of like prokaryotes. They're kind of like eukaryotes. Don't really know much about them. That's it. What? What do you give me a whole page of information for? That's the thing, right? Whereas just like, also, neurons and muscle, we will have our entire chapter on, right? If you guys are the read ahead type of people, you ready for this? I don't know if you guys have ever seen me do this before. Neurons are in chapter 4, and muscles are in chapter 11. And if you don't understand why that's so special, remember that there's 4 books that I teach, and each of them have 12 chapters. So it's 48 chapters total, and I know where every single piece of information is in every single book. Not because I'm smart, but because I've done this a lot. Classifications of bacteria. I'm going to let you guys do this one. Sphere-shaped bacteria are called cocci, or cocci, right? Rod-shaped, bacilli, and spiral-shaped are spirilla, right? There are certain names of bacteria that you'll be able to see, and this is important for like passages and things like this. So if you see Lactobacillus, Acidophilus, Bacillus, the bacilli. S. aureus stands for Staphylococcus, right? Does anyone know the spiral-shaped bacteria? Anyone know one? It's going to blow your mind. It's one of the oldest bacteria known to men. It has a very simple treatment, but a lot of people die from it because they don't get it treated. Or a lot of people did die from it. Huh? No. Measles is a virus. MMRV, right? No. Streptococcus. Treponema pallidum, also known as syphilis. What is the treatment for syphilis? What is the treatment for syphilis? You guys will be blown away. You think it's some crazy meropenem, carbapenem, crazy doxycycline, like 4 weeks of this, 4 weeks of that. One single shot of penicillin. One. If you have primary syphilis, you get a shot of penicillin and you're good.
What would you do if you're allergic to penicillin?
You desensitize the patient to penicillin. You give them tiny, tiny, tiny doses of penicillin to lower their allergic response and then dose them. And you do that in the hospital. I had a patient with tertiary syphilis once. Means that the syphilis got into their bloodstream and was entering their brain, right? So the normal thing for syphilis is one shot, which is, I think, 1 million units of penicillin G. Penicillin G is a specific type of penicillin that you can inject. The treatment for tertiary syphilis at the time is 15 to 21 million units of penicillin G every single day for 14 to 21 days through an IV. People should get treated when it's early. Very interesting stuff. Aerobes and anaerobes. What is an obligation? Everyone, if I said it is obligatory for all the men in this room to come eat tacos with me later. What is an obligation? You must do it. The prayer we did before, that is an obligation. I cannot miss that prayer. It's not possible. It's not feasible. It's not allowed within the Islamic code of law. Within the code of law, if you are a man, there are acceptances and allowances for people who are sick or in a coma or sleeping or forgetful or this or out of their state of mind, or many, many, many for women who are on their cycle, right? Or pregnant labor, this, that, whatever. There's so many allowances. Islam is a religion of mercy before anything else. It is a religion of intention and a religion of mercy, right? However, if you're a healthy man and you don't have any excuse like sickness or coma or whatever it is, and you're sane of mind, you must pray 5 prayers every single day. You have to. I had a friend who asked me one time, he's just like, why do you do this? I was like, I have to. He's like, what do you mean you have to? I was like, I have to. There's no ifs, ands, or buts about it. In the same way that you have to drink water, I have to pray. No ifs, ands, or buts. No questions asked. So when you talk about obligate aerobes, they have to have oxygen. Need oxygen. And when you talk about obligate anaerobes, oxygen would kill them. Yes? How does oxygen kill? How does oxygen kill? There's a very interesting phenomenon known as radical oxygen species production. You know all of those words. You know what oxygen is, you know what production is, you know what radicals are. Put them all together. if you want to do the in-depth reading, if you want to learn something a little special, look up reperfusion injury. And you guys will see just how toxic oxygen can be. Not to breathe, but you know. A facultative anaerobe will swap to anaerobic processes when they are beneficial. Give me an example of a facultative anaerobe. Oh, you're looking at one. You're looking at a facultative anaerobe. I am currently undergoing anaerobic respiration. If you put a noose around my neck and made me run a marathon, my muscles would swap. Aerotolerant anaerobes. Anaerobes that tolerate air. They don't use it. They're like, it's fine. Yes? Is everyone up to speed? Everyone's good? Okay. You know what I love about this MCAT course and teaching it? Is that in the beginning, you see everyone, they're just like, yeah, we're good. We're okay. We're fine. And then we get to chapter 2. And I have to tell you about how the progesterone withdrawal within the female's body leads to an increase in gonadotropin releasing hormone from the hypothalamus, which increases gonadotropin release from the anterior pituitary gland, acting upon the follicles and ovaries of the woman in order to increase the deciduization and endometrial growth in preparation for the new menstrual cycle that will actually negatively affect the GnRH to grow the single follicle that's most sensitive to the estrogen that's being produced. And then the estrogen that's being produced actually floors out at a specific level in the ceiling, which turns off that negative transmission to the hypothalamus. You get the LH spike, which allows for ovulation. And that's chapter 2. We'll get through it. I promise. So we'll do that on Sunday. If you're scared, sleep in that day. But even if you sleep in, I'll be here at 4:30. Prokaryotic cells. Before all the estrogen, progesterone, GnRH, everything, blah, blah, blah. Also, it's so weird teaching this course. It's just like, especially like, I was teaching to a smaller group in Brooklyn College. Like, I feel more comfortable now because like Brooklyn College is a smaller group. I think I could literally see it in front of me. Like it's like 4 people or so the first time I did it. So my second chapter was literally just all about the menstrual cycle. And there was like 3 women in the room and 1 man. And I'm just standing there talking about like periods and the menstrual cycle and pregnancy and this and that. And I'm just like, I don't know anything about this. What am I doing? I felt like such a poser, you know what I mean? I felt like just bringing like my sister with me, just be like, please help. Oh man, we'll get through it. It's very complicated. I'm not gonna say that it's not. It's super, super complicated. Okay, so prokaryotic cells, basically they have some things that eukaryotic cells don't. They have a cell wall, right? And this cell wall either has a thick or a thin peptidoglycan layer. A thick or a thin peptidoglycan layer. Okay? This is gram-positive. And this is gram-negative. You're staining the peptidoglycan. That's what you're staining. Right? And I want you guys to remember one thing as you journey into medical school that will stick with you when you study microbiology. It is very important to understand that the gram-negative bacteria actually cause much harsher infections because of one thing. It is known as lipopolysaccharide, or LPS. Guys, when we get sick, what is it? What is making us feel sick? Don't say an infection.
The immune system.
It is your immune system's response to the infection. Lipopolysaccharide is an incredibly immunogenic molecule. It activates the immune system like pouring gasoline over a fire. So your immune system lights up and flares up, and your blood pressure drops, and your heart rate goes up, and your temperature spikes, and you feel like crap. Now I'm talking about very severe infection. I'm talking about sepsis. Sepsis is a very specific term. I don't use that lightly. Okay? These prokaryotic cells often have flagella. There's a beautiful diagram of the flagella in chapter 1 of the Kaplan book. You can look at it. And what happens is that the filament of the flagella and the tip of the flagella is able to rotate in such a way from the basal body, right? The basal body generates such a force that it whips around, and because of the long arm, the moment arm of the flagella, it generates torque, and that torque allows it to propel forward, right? Does that make sense? Beautiful, beautiful stuff. Very good. Is everyone following? Who here is tired? Be honest, who's tired? We're almost done, I promise. I know I'm keeping you guys here for a long time, but it's for your own good. For your own good. My students who did the best on the I remember. I'll actually tell you right now. I remember 3 of my students who came to every single one of my lectures at Brooklyn College. I will not give you their names, but I will give you their initials. These are their initials. They came to every single lecture I had. He got a 519. She got a 517. She got a 516. They were my best students of the first batch of students that I had. Stick with me. I've never had a bad review from a student. In 3 years, I've never had someone say that it wasn't worth it. I take pride in that. I take a lot of pride in that. But be cautious. Do not get cocky. I had one student whose full-length averages was a 523 average on their full lengths. Across 5 of them was a 523 and went in and got a 512 the day of. Tragic. Do not get cocky. Prokaryotes also have a nucleoid region. Because they want to be us so bad. It's like, oh, I can compartmentalize. I got this. No, you don't. Stupid, idiot bacteria, dumbass. And they can also transfer and acquire DNA through plasmids, which are little rings of DNA that are separate from their actual DNA. The nucleoid region is where they kind of just hold their genetic material. It's a general location. Correct? Yes? And we will talk about this now. The ways that prokaryotic cells reproduce. Number 1, do the SpongeBob. Binary fission. Do you guys remember that episode of SpongeBob? Who here is a big SpongeBob fan? Like I am. I love SpongeBob. Love that show. It's my favorite show ever, of all time. The first 100 episodes of SpongeBob are some of the best TV ever written. That and House MD. Oh, and Attack on Titan. Attack on Titan is pure, absolute cinema. Peaky Blinders, great show. Fantastic show. Jahil, definitely like very, very raunchy stuff, but great show. I watched it like 2 years ago. What else? We'll talk. Who remembers that episode of SpongeBob where he's talking about the hash sling slasher? And he's like, and he cut off his own arm. He's like, you mean like this? Like this? And he keeps pulling his arm off and it keeps multiplying because he's a sponge, right? Binary fission is basically that. The bacteria is literally just like, watch this. And he just starts breakdancing and now there's 20 of them. That's basically what happens. That's binary fission. Imagine if I walked out the door and came back and there's 2 of me. You guys would all be running for the door. Not only because it's weird, but because imagine a world with 2 of me. Oh, what a great place. I hate myself. Genetic recombination. You guys will never truly know how I feel about myself. That's one thing you'll learn in this class. Sometimes I will say I'm like the dumbest person, most ignorant person in the entire world, and then I'll be like, yo, if there were 20 of me, it'd be lit. I don't know if I mentioned earlier, but I have a degree in philosophy. That's what I got my undergrad degree in. So that's why I'm so conflicted about all my thoughts about everything. I've spent so much time thinking that that's all I do now. Anyone here ever dabble in philosophy? Either on your own time or in school? Yeah? Yeah? Cool. Very good. Let's talk sometime. Genetic recombination is the method by which bacteria are basically able to modify themselves and modify their genetic material without actually needing to have sex. Because when humans do that, they have to have sex, reproduce, pass on genes, and evolve over time, right? Bacteria sex is a little bit different. Guys, we're going to be very, very, very straight up about everything that happens in this class. We're going to talk about periods and sex and intercourse and fertilization and death and dying and bleeding and this and that and the cardiovascular system and fetal development and everything. We're going to be very upfront. Very, very upfront. Humans, in order to pass on genetic material, need to have sex. And sex is a very energetically expensive thing to do. You use a lot of calories. You can only really do it when you're healthy, right? Et cetera, this, that. And it has consequences, right? So what bacteria can do is they can actually just take up genetic material from around them, from other bacteria, and from other things, right? So let's talk about these things that they can do. Number one, it's called transformation. Transformation. This is when a bacteria dies. You have a bacteria with all of its plasmids inside of it and its DNA in this big coil, right? And when this bacteria dies and it releases all of this stuff, another bacteria with its DNA is like, hey, I want that. Just picks it up. Transformation. Make sense? Following? Yes? Tired? Yes, we are. Yes, we are. We are tired. I got to school at 8 a.m. this morning. I have not taken a break since. I am also tired. I'm right there with you. I am your friend. Even though I'm your teacher, I'm your teacher who is your friend. But I'm not only your friend. That is a lesser title than being someone's teacher. Conjugation. I meet a lot of people in New York who see me in public at a coffee shop or a bookstore, whatever it is. Like, oh my God, I watch your videos online. Happened to a couple people in Dallas as well, right? Like, I think at this point, like 6, 7, oh God. People, like people who have approached me and been like, oh, like I know you from your videos, things like that. Oh, I shouldn't have done that. I should have just lied. Shouldn't have said anything. I'm gonna hang myself. No, I'm not. No, I'm not. Guys, I'm okay. Don't call anyone. I'll be okay.
I'm gonna clip this.
Huh?
I'm gonna clip that.
It's already clipped. It's on my YouTube. Everything I've ever said is clipped.
Can you hit the email for us?
No. No way. Off camera, maybe. Okay. What was I saying? What was I talking about?
Something about meeting people.
Yes. And yeah, but I forget where I was going with that.
They watch your videos and you meet them like in person.
Oh, right. So I meet them in person. It's just like, people are just like, oh, like, I didn't know if you wanted me to say hi or whatever it is. Guys, I love meeting people who have benefited from my content. It is the most beautiful thing ever. Because this, to me, is my life. This is what I love to do. This is my passion. This is what I do with my free time. I love this stuff. And the fact that things that I love to do make people's lives better and easier, oh, nothing better than that. I'm living the life. Who could ask for more? I eat and I sleep and I live in a nice home with nice people and I have a good family, alhamdulillah, and I study and I work, and people benefit from what I do. What could be better? Who's living a better life than I am? Seriously. And I want this to be you guys. That someday you're going to be in my position, you're going to teach. It's going to feel great. going to feel fantastic. On that note, let's talk about bacteria sex. What's this one? Conjugation, yes. There is a F plus bacteria that is able to make a bridge, and an F minus bacteria that is unable to make a bridge. And that ability to make a bridge is actually coded within the gene that they're passing on, and other genes as well, right? So let's say that you had a bacteria with a main DNA, and you had F plus and one called vanc. And you have another bacteria with just this DNA. The F plus gene inside of this bacteria allows it to throw a bridge. This is called a conjugation bridge. This is bacterial mating, correct? And over this bridge, it is able to copy and transfer these plasmids. So now this F minus bacteria becomes an F plus bacteria with vanc. And it is able to go to other bacteria. Let's say that this one is marrow, and it can throw a conjugation bridge. And not only can it give vanc, it can accept marrow. And now these two bacteria, because of this one, can mate and have vancomycin and meropenem resistance. And then bacteria just start tossing lines over to one another. And that's how we garner resistance, right?
Can an F1 throw a bridge to another F1 or is there only
Yes, 100%. If you have the F, that's a very good question. Question was, can an F positive bacteria throw a bridge to another F positive bacteria? 100%. Once you have the ability, you can. Okay. We are going to go a little bit over time. If you guys need to leave, it is almost 9 o'clock. If you guys have a long commute home, please go home. Being very serious. Now is a good time to leave. It's not much after this, but we are going to go over time. Okay. Some plasmids can be inserted directly into the main line of bacterial DNA. Just remember that. They are known as episomes. Episomes. Okay? Transduction is basically when you have a virus. Like we said, these stupid, dumb, idiot moving packages of genetic information that will sit on a bacteria and inject this DNA into them. Yes? That's it. Transduction. Good? This next part, I used to take a lot of time explaining. It's very straightforward. I feel like in the past, the one mistake I made is that I underestimated my students. I thought that they were all as intensely stupid about everything as I am. I'm very dumb about things I've never read. You will find that I am critically stupid about a lot of things in this world because I've never encountered them. So I need to do everything from the very beginning. But not you guys. You guys are probably a lot smarter than I am. What is this? It's a bacterial growth curve. Yes? With time accelerating and the log number of colonies. We have a lag phase, an exponential phase, a plateau phase, and a death phase. And this actually goes all the way down. Bacteria are grown in an environment with limited resources. if you don't let resources into an environment, everyone's going to die. I will say that again. If you do not let food and resources into an environment, everyone will die. Okay? Once the bacteria eat up enough of their resources and they're not able to sustain this growth anymore, they plateau out. Because the rate of growth equals the rate of death. The ones that grow, they eat. The ones that die, die. And it happens at an equal rate. We've reached an equilibrium here. This is a very beautiful example of biological equilibrium. We always talk about chemical equilibrium. This is biological equilibrium. But once they've established themselves, boom. Takes a little bit of time, but boom, right? And once they've plateaued, they eat through all their resources, and they die. Okay? Very straightforward. Anyone need that explained? Cool. Viruses and subviral particles. Good thing I still have a picture of a virus right here. I don't love it. We'll do it again. They just look like little aliens. Capsid. Protein coat that protects it from whatever it needs protection from. Idiots. Some envelope of lipids that you see over there. And just some genetic material. Could be circular, linear, single-stranded, double-stranded, RNA, DNA. They don't care. They do not care. Right? Someone named me a very famous virus from the past 100 years. COVID. Okay, next. HIV. Next. Flu. Next. Measles, rubella, CMV, HSV, HPV. Viruses are everywhere. They've been here long before we were advanced as a society, and they'll probably outlive us as well. Because they don't actually have to live. They're not going to outlive us. They're going to out-exist us. Right? Okay. Viruses cannot reproduce by themselves, but they need obligate hosts. And with these hosts, they are able to take and inject their viral material. I want you guys to follow this next step very closely. I was wondering who I picked this up from, this little thing that I do when I want to explain something. It's from Marco Pierre White. Have you guys seen his videos? Marco Pierre White's a very famous chef, and when he looks at the camera, he says, "Listen to me very closely." And I picked it up because I think it's very poignant. Poignant? I don't know. Poignant, right? It's a very poignant thing to do. So, listen to me very closely. The DNA/RNA, and if it is DNA, it gets turned into RNA, correct? Yes? Will eventually make proteins. And those proteins will go to make a viral capsule and viral materials. And you take the genetic material, the viral capsule, the viral materials, you put them together, and you get a virus. So by injecting its material inside of the cell, the virus actually creates a space within which it can make more of itself. Yes? And this will tie into the lysogenic and lytic cycle. Yes? They lack ribosomes. They need some host cell to replicate. They can't do anything by themselves. They're like babies, right? They're like children. We'll do the whole fetal circulation thing. Have you guys seen my fetal circulation video? You guys remember that rant I went on where like, this baby is a dumb, stupid, useless, crying, idiot parasite. Why? Because all it does is mooch off mom. That's it. If we didn't have the literal like sort of biologic attraction to babies that we did, the human race wouldn't exist. Right? People in the back, is it difficult for you to read? Do you want to move forward? Take a couple seconds, move forward. There's a lot of empty seats over here. I will stop writing for a few seconds.
Have you seen the newborn effect? Where moms are like convinced their newborns are like the cutest thing ever.
We will explain why the newborn effect happens on Sunday. Literally on Sunday. I have a scientific explanation for why it happens.
Oh, what does it say next to viral capsule?
This?
No.
Capsule and just viral like materials.
Doesn't the newborn effect happen just because of a crash of oxytocin? Or just like
A spike, a massive spike of oxytocin. It's one of the theories as to why.
Oh, it's theoretical. It's not like a known.
Nobody knows. You know what's one more thing no one knows? Guys, hold on. You guys know what's something that nobody can explain yet? Why labor starts when it does. No one knows. Absolutely no one knows. It is a scientific miracle why labor starts when it does. We have theories. We're like, oh, neural development, this, that, the other thing, signaling molecules, blah, blah. Nobody knows. We have theories means we have no clue. Bacteriophages are viruses that specifically target bacteria and perform bacterial. Wrong. Again, specific. Bacteriophages are viruses that target bacteria and perform bacterial. Louder. Very good. Transduction. You need a bacteriophage in order to do transduction. Wait, did we go over transduction? Did I skip that? Oh, okay. Okay, good. I remember I had another diagram. I was like, what the hell? Come on, guys. Himma, drive. Let's do it. Okay. So we talked about single-stranded, double-stranded, blah, blah, whatever it is. Now, one thing that the MCAT loves to write passages about. Not that they will specifically ask you a discrete question, but they love to write passages about. What are retroviruses? They are not viruses from the 70s. Although, we'll get there. Retroviruses do what?
Do they need their RNA reverse replicated?
They take their sensitive RNA and use, what's the name of the enzyme? Reverse transcriptase to produce DNA, hijack your cell, slip its DNA into yours, and hijack your entire cell's wiring to make more of itself. And the reason I made the joke, but it's not really a joke, it is true, of a virus from the 70s is because what is a very famous retrovirus? HIV. More the 80s and the 90s, but you guys got what I mean. Oh, HIV. Scary stuff. Scary stuff. Don't be stupid, guys. Please. Okay? I want someone to make a note that we will speak about, you know what? I'm going to leave it up to you guys. We can speak about the lysogenic and lytic cycle. It will take 15 minutes, 10 to 15 minutes. Or we can leave it for when we do immunology. Immunology is a very, very complicated lecture. Only takes about an hour and a half. Very short lecture. That's very short, by the way. Short lecture. We can do it at the end of immunology, but someone needs to remind me because I will forget. Okay? You guys want to go home? Let's go home. Okay, one last thing. One last thing before we go. What is a superinfection? A superinfection is a sickness, bigger for the people in the back, a sickness over another. Staph aureus is a common cause of post-influenza infection. Post-influenza infection, Staph aureus causes it. Influenza shoots your immune system, weakens it in the meantime because of the amount of work that it's doing, and it breeds the ground for the infection with bacteria. That is a superinfection. 2 more points, and we're done. Prion disease. What is the most common prion disease that you read about in medical literature? Creutzfeldt-Jakob disease. Creutzfeldt-Jakob disease. It is known as CJD. Creutzfeldt-Jakob disease is a prion disease. What is a prion? A prion is a misfolded protein. It folds incorrectly. And when it encounters other proteins, it causes the proteins to misfold incorrectly in the same way as itself. And it hits other proteins and it does the same thing. And it hits other proteins and it does the same thing. And they stack 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 until they block up your body. Creutzfeldt-Jakob disease is incurable and 100% fatal. It is a brain disease. It causes a massive, rapidly progressing dementia. And there's 2 paths by which it can happen. Spontaneous, meaning it just happens, which is exceedingly rare. Exceedingly rare, right? I have heard of neurology attendings who have been working for decades who have seen 1 case. Or ingestion of the prion. Undercooked meats, eating brains, eating humans. Don't do that. Don't put yourself at risk. And one last thing. Oh, I'm tired too, guys. Viroids or whatever it is. I don't know. Viroids, I forgot. I can't even read my own handwriting. Viroids. They are tiny viruses. And they infect plants. We do not care about plants. You do for a biology degree for whatever reason. You do like plant bio. But we don't care about plants. Not here. Any questions? Thank you guys so much for coming and listening. And I hope that you benefited from today's lecture. I will see you all on Sunday at 4:30. Thank you. Please turn off the camera.